Travis is going to be our first speaker. He's a senior lecturer at La Trobe University who grew up exploring the waters of Western Port. He's a physiologist by trade, but has varied research interests from the feeding behaviour of pelicans to the effects of high-intensity exercise on the contraction of human muscle fibres. That sounds like a perfect recipe for someone to talk about elephantfish. He's also part of the research team that successfully had the bryozoan reef community in Western Port listed as a threatened community under the Flora and Fauna Guarantee Act. And that has only happened very, very recently, and it was due to a whole of amazing scientific work, and that could basically be a whole nother presentation, but feel free to ask him questions about it because he does love talking about it. Tonight, however, he's going to talk to us about the elusive elephantfish. So it gives me great pleasure to introduce Travis Dutka to share his screen and start to entertain us. Thanks, Travis.
Thank you, Cade, and thank you for the opportunity for me to give a presentation wearing my tracksuit pants. I think it's that kind of weather. A hidden gem. Well, the elephant shark really nails that brief. Not many people interact with elephant, shark, fish, whatever. I'll explain to you what actually is a chimera. So basically it's got a lovely name, actually, the genus. It means it's Calorhynchus, and calo meaning beautiful and rhynchus meaning snout in Greek. So beautiful snout. And the chimera have a whole range of different morphology of their snout. The one that I'll be talking about is the species known as Calorhynchus millii, and it comes into the embayments around the southeastern areas of Australia. So just a little bit of background about them. They separated away from sharks and fish about 400 million years ago. So there's three lineages of fish. There's the cartilaginous sharks and rays, then there's the chimera, and then the bony fish, the true bony fish. They fall in a subclass called holocephali, and the family chimera, they shared, or the family's Chimeridae, but a chimera shares traits with both sharks and bony fish, truly bony fish. So they're a bit of a hodgepodge of traits, and I'll go you through some of those quirky ones, and then we'll really get to the really interesting ones in association with their reproductive side. So there's 5 genera in total and about 47 species globally that we know of. There's probably more. They're found typically in the deep sea environment, which is very hard to explore and get a really good grasp on specimens. So the unusual thing about the genus Calorhynchus is that they spend most of their time in the deep sea, but then they traverse into really, really shallow water. So they make this quite an unusual journey, which is not just typified by a long travel, it's a change of habitats. So they go from beautiful, clean, deep sea water to murky, shallow, muddy water. And this occurs in autumn when they come in to lay their eggs. So Calorhynchus millii, the Australian ghost shark, can be found in Australia and New Zealand, but there is other very closely related species, Capensis, which is found in South Africa, and another species, the name escapes me, found off Chile. So they are found in the southern regions, and they typically reside in about 200 metres, but then in autumn they can come up into about 1 metre of water to lay their eggs. So just some of the traits which I'll run through between a shark and a fish. So these traits that the chimera have, one thing is the skin. We look at the skin. So most of you would have felt at some stage some shark skin. So it's smooth if you rub your hand in one direction and really rough if you run it the other way. In fact, shark skin was used as emery boards for polishing nails and really fine grade sandpaper. And if you look at them under a microscope, you can see they've got these jagged teeth. So they're called dermal denticles or skin teeth, okay? The ghost shark doesn't have that. It has skin more aligned to a bony fish. I won't go into too much physiology, but basically Chimera and your sharks and rays are what we call osmoconforming organisms. So basically, when the salinity of the water changes, they change the number of particles in their cells to maintain equilibrium with that environment. So if the salinity increases, the salinity of their internal fluids will increase. So they conform to the change. Whereas the bony fish, they have the completely separate approach, and that's to regulate in spite of environmental changes. So if the water becomes diluted with rain like this, or the seawater is concentrated in rock pools and whatnot, they maintain a constant number of particles in their cells, and they deal with the change instead of going with the flow. Chimeras have a low reproductive rate, which aligns more with the sharks and rays. So over a breeding season, a female ghost shark can lay probably about 20 eggs. But whereas some fish, like a flathead, might liberate, 1 million eggs, which can be fertilized externally. They swim actually through the water in a quite an unusual way. So most fish swim in a serpentine motion where they move the spine and tail beating like that, whereas these guys keep their spine straight, and it's a really tapered body shape that they have, and they have their exaggerated pectoral fins on the side. They flap like a bird, okay? So they use them like wings to fly. They fly through the water. And if you want to have a look at this, you can YouTube elephant shark swimming and you'll see this. Just some other, just moving that bar out of the way. They have one gill opening, which is like a fish. So fish have a gill plate called an opercula, which sits over their gills protecting it. And they have one opening or one slit, and elephant shark have the same. Whereas sharks, this is a 7-gill shark for obvious reasons. You can count them up here, but they have multiple openings for their gills. So that's another trait that sort of distinguishes them from a shark. So they have, chimera males have claspers, just like other sharks and rays. So they're reproductive appendages, which are paired and sit outside the body. And this is one of the first things to identify the sexes of a shark and ray. You can easily see these structures, and that can tell you straight away underwater whether you're looking at a male or a female. But it gets even weirder than this. The elephant sharks have taken sort of sexual dimorphism to an extreme. And if we look at some of the other traits that they have, sorry, for my screen, I have to keep moving the bar out of the way. So if you look in front of the pelvis, you'll see here they've got these pre-pelvic pouches, and sitting within that pouch are what we call pre-pelvic tentaculum. So tentaculum normally means tentacle, but in chimeras it means a reproductive clasper, a supplementary clasping organ. And when it's sitting and swimming, it has it tucked away neatly. But when you actually manipulate that, it comes out of that pouch, and you see this weird and wonderful structure, and it's got these gripping pads which they use to hold onto the female during a coitus. Not only do they have those weird structures, they have a weird protuberance that sits on top of the forehead, and these are called and I'll show you, it's reseated here to make it streamlined for swimming, but when it needs to, it can actually pull that out and use that as a gripping tool to latch onto the female. So this is called a frontal tentaculum or frontal teeth. And if you can look at the actual higher resolution, you can see all of these points, little spikes. So it's really like a little club which it can use and grab hold of a leading edge of a female's pectoral fin or her dorsal spine. So these sharks have a spine sitting on top of their back. And when breeding season comes about in autumn, usually between the end of or middle of March through to the early parts of May, the longer the breeding season goes, the more scarred you see on the female. So this is a female now who's been mated with or attempted to be mated with, and you can see all these gouge marks along her body where the male has tried to grab hold of her. And we know that these are not injuries from any other cause, because if you look in these little yellow boxes that I've got circled here, you can see they fit that club-like structure on the head of the male. So what I think happens is these long scratches are caused when it tries to grab hold of, but she either wriggles away or he slides down, and that causes these longer scratches. So very little is known about the breeding behaviour of the Calorhynchus, the actual act of mating, because it's usually in murky water and people just don't see it. So we're trying to understand in our laboratory. So I've combined my efforts with the neuroecology lab at La Trobe Uni, and together with Sean Collin, we co-supervise some PhD students, and we've got a talented one called Hope Robin, who's working on the sensory modalities of these species of Chimera. Nothing is known about the sensing abilities of these and how they cope with changes in environmental conditions, how it goes from clean water to dirty water. How does it rely on its vision and then switch to some other cues? There's no understanding of how well they smell, how well they see, or how well they hear. So here she's dissected painstakingly out an intact brain of an elephant shark, and you can just see how elaborate the structures are. I'm not going to go into all of these different parts of the brain. That could be a whole presentation in itself, but I just want you to appreciate that they're not just a simple P-shaped brain with not much going on. They've actually got a highly developed brain, like most vertebrates, and all these regions relate to a particular sense. But they're quite interesting, and in the next year or two, we should have some lovely answers in the behaviour and the development of the senses in these animals. So they're rarely seen by divers and snorkelers, partly because 10 months of the year they're not here, and the 2 months that they are here, they're in murky water. So, and typically they use these areas of murkiness to lay their eggs into the sediment, and you can find these guys in the upper reaches of Anderson's Inlet, Western Port, to the east side of Western Port in particular, and to places like the top of the Barwon River as well. So for 2 months of the year, they come in and they reside in poor visibility water. But if you really wanted to spot one in the wild, I'd suggest you go at high tide when the clean oceanic water pushes into areas of the eastern side of Western Port, around Grantville and Lang Lang, and just snorkel over the flats in about 2 to 3 metres of water, and don't kick vigorously, and just maybe do a drift dive or snorkel with the tide, so you can actually not startle them, and that's probably your best bet at seeing them. So they come into these regions and they lay pairs of eggs. So each time the female lays, she lays 2 eggs in soft sediment, and she can lay up to 20 per season. We don't know whether she makes the same trip every year or whether there's a hiatus of about 5 years or so, like some other sharks can do. And then usually in migratory animals, it's sometimes a challenge to do it every year. So we don't really know too accurately about the fecundity of these animals. And these photos are courteous of by Rudy Curto. If you've into your fish identification and you've looked at any fish book, chances are the photos were taken by Rudy. He's ubiquitous in the Australian dive world and snorkeling world. So, as I was just saying, most people who encounter them are fishermen, and they can actually target them in these areas. What I think, so in the 80s and 90s, there was lots and lots of elephant shark caught all around the entire bay. They imposed bag limits to reduce the catch down from 5 down to 3 down to 1. But I think in most of these areas where people are familiar with, they no longer catch elephant shark. And alarm bells went off, and we thought that perhaps there's fewer animals around. But the fishing status is stable, and the numbers being caught out in the ocean are seemingly unchanging. And I think what's happening is, in the past, as the seagrasses recovered in Western Port, the Zostra, there's less and less area for these animals to lay their eggs. They need bare sediment. So if the Zostra is doing well and recovering across the bay, they're being forced to utilise the remaining small patches. So I think there's a contraction of the area that they use, and the contraction's not in their numbers. So I think their numbers are probably stable. They're just utilising different regions now. So if you really want to encounter these guys, you probably need to go to the eastern side of Western Port. Now, just a quick close-up of their face. You can see they've got these weird crushing teeth, and they use these to break the hard bodies of crustacea and bivalves. They eat mud arcs and whatnot. They have lots of electroreceptors on these snouts, and the snout morphology changes from species to species depending on the evolution of its interaction with its preferred prey. Another unusual trait that they have is they have their nostrils inside their jaw, so the nostrils go through the jaw, which is quite unusual. This is just a picture of some of their preferred food. So they come in and they graze in the shallow sediments looking for ghost shrimp, and by little bivalves, they crush them up with those strong crab and shellfish designed jaws for crushing. So this is just a picture of taken from April this year at Lang Lang. We call it a boat ramp, but it's just a slab of concrete in the mud. It's getting a bit of an upgrade, but it's not much. So this is a very expansive, shallow, warm, bare sediment area, perfect for their eggs and the developing embryo, because the shallow water with less current actually has probably a higher temperature to it. So it's probably conducive to developing embryo. And here's some, so fishermen here are either targeting gummy shark or elephant shark at that time of the year. And they do so with kayaks because they can access the shallow water. So there's not many people who actually get to interact with one in the wild unless you're a fisherman targeting them in a specific area. So that's all I want to say, and I'll hand over now. Back. I'll stop sharing, maybe.
Thanks, Trav. That was fascinating, and we've actually got a question here from Dolphin Dave Donnelly, who doesn't know how to use the Q&A function. But his question is, how do the gill openings of the elephantfish differ from the spiracle of a ray?
So the spiracle of a ray is part of, because they're a sedentary animal who are sitting static.
Actually, can you tell us what a spiracle is? Because some people may.
Okay, it's an opening. It looks like an ear, but it's not. It's just a big hole. If you swim over a ray or like a fiddler ray, banjo shark, or any skate ray, you'll see they've got these large openings behind their head, and they're called a spiracle, or it's a buccal pump where they draw water in, okay, under pressure. So because their gills are on the underside, and if they're sitting sedentary on the bottom, they're not pushing water over their gills. So when they're sitting idle on the bottom, they use these buccal pumps to suck water in and push it over the. So these guys don't do that. They've got their opening to the side and they're drawing in water through the mouth and poking it out through that opening. So that's the difference between a buccal pump, or spherical is the opening, but all taken in its entirety, it's a buccal pump.
So I've got a quick question here from someone who might have missed it, and I wanted to just add to it. It's like, so what are the 2 months, and is this at the top of the temperature in the bays, or is it sort of as it's starting to dip off?
Yeah, it's starting to dip off, but it is. They come in maybe the end of February and leave by sort of mid-May. But each individual is usually only in there for no more than sort of 3 months, but typically about 2 months, and it does coincide with warm water. So,
good tip for those wanting to find it. So
yeah, I would say middle of April, if you're looking to swim with one, pick a high tide, shallow water, either Grantville or Lang Lang, when you got clean visibility that time of year on a high tide. Yeah.
And so what you're telling us that having it on the fish count slate is pretty much irrelevant because no one's going to find it in November or December. So
unless they find a juvenile that's
okay,
hatched and it's residing in the bay.
What I wanted to know is this: you talk about the soft sediment. So soft sediment is a very catch-all phrase for sediments in general. Is there, like, do we know, is it a particular sort of like a clay, a sediment, a sandy, like, do they lay it on a certain thing? And then, often most people's interaction is with the egg case itself. Is that just a shark that's laid it in the wrong spot, or is it usually after the juveniles have hatched?
Well, a bit of both. I think once the juvenile is hatched and erupted out, it becomes light and dry, and then probably makes it more likely to be disturbed by waves and whatnot and current. But also, if there is a storm activity, you can find viable egg cases on, and same is true with Port Jackson shark and fiddler rays and a lot of other rays and things. They can be viable, but a storm surge might have pushed them up into the shallows or up on the beach. So, yeah, don't take them home. Just have a look and see. You can actually candle them. It's what the technique. If you shine a light through, you can probably see if it's got a viable embryo in it, and then you can put it actually back. And in terms of the sediment, that side of the bay all changed dramatically about 100 years ago when they drained the Kururup swamp to make it agricultural land. So I think the sediment load is probably clay and soil that's washed down, and also from erosion from that side of the bay. Whenever there's a storm activity pounding into the banks, it erodes and it deposits and spreads it down. You're on mute, Kate.
I've got a kid that's just gone to the toilet, so I didn't want everyone to hear that. You can use the soap bud, it's fine. We've had a power blackout. Yes, we can get a new one. We've had a power blackout. So Mike is hoping that it's going to come back in 5 minutes or so. We've also lost Nicole, so I have a feeling that it may be widespread. We have most attendees. Nicole, well, back, and now she's gone, so Nicole is still here. So I've actually just got a couple of questions, and if anyone's got questions for Trav, please just throw them in there while we're going. So you've answered my one about sediment, and now you're talking about like the reproductive behavior and not really knowing much about them. Are they a species that doesn't keep well in aquarium settings? Like, is that something that's been tried?
They were for a while, and Dave Donnelly probably can speak more to this. They were a feature of the Melbourne Aquarium for a few years. I'm not entirely sure as to why that disappeared. I don't think they do very well in captivity, and I think certainly they need probably tanks which don't have corners and whatnot, in if it's a circular shape, that probably makes it easier for them. But again, we don't know much, if anything, about their sensory capacities, what irritates them, what not, how sensitive they are to noise, how sensitive they are to, electrical discharge. There's lots of reasons why they might not keep very well in captivity. So we'll hopefully over the years we'll understand a bit more about their physiology and how that influences their behaviour.
Yeah, don't worry, we'll be inviting you back. There's been a lot of good comments and people sort of saying keen to go exploring now. So it'll be interesting to see the snorkelers with the kayak fishermen. Michael is the manager of Marine and Coastal Sciences and Programs at Parks Victoria. Prior to joining Parks, Michael spent oodles of time in Port Phillip Bay getting to know marine sessile communities. If you want to know about sponges, bryozoans, ascidians, Michael is your man. I don't know if that knowledge is still there, but I think it would be one of those things that would come back to him if he was put in front of a settlement plate. He also learnt a lot about tropical rainforest plants while participating in a study that ran, and I use the word ran, 1,500 kilometre transect. So any of you have done any science, basically everything's done with a 50-metre tape measure. This was just a little bit bigger along the east coast of Australia. So he's actually moved from the sea to the trees, basically. He's also got really good knowledge about tropical rainforest plants as well. So again, gives you a good idea of how diverse our speakers are tonight. He's also spent some time hooning around the bay in boats while working for the EPA. And he's been in his current role for 18 months and was gifted with the task of wading through over 20 years of research done in Victoria's marine national parks and sanctuaries. And tonight he's going to do his best to condense that into about 10 or 15 minutes. Gives me great pleasure to introduce Michael Sams. Thanks, mate.
Thanks for the introduction, Kate. Yes, bryozoans and sponges aren't things you forget. I'm a bit rusty, but they're still pretty burnt onto my brain. Yeah, look, thanks for joining me, everyone, and thanks for the introduction, Kate. To begin with, I'd just like to acknowledge the traditional owners, the First Peoples on the lands we're meeting today. So I'm beaming to you from Wurundjeri country, very wet Wurundjeri country right now. So I'd like to pay my respects to their elders, both past, present, and future. So as Kate said, I just want to talk to you about some of the monitoring and research that we've been doing at Parks Victoria over the past 20 years we've had with marine parks. And before I get into that, I should say that most of this research has been done with research partners at Deakin University, Melbourne University, and universities across the state. So I won't be able to acknowledge them through the talk, but just keep that in mind. Before I get into that, though, I just want to take a step back and talk about Victoria's marine national parks and sanctuaries. So around 20 years ago, Victoria created a network of highly protected no-take marine national parks and sanctuaries. And so the purpose of these was specifically to protect representative examples of Victoria's marine biodiversity and the ecological processes that support that biodiversity for future generations of Victorians. So many of you will be familiar with it, but this is our network here. So our system of marine protected areas spans the entire state from Cape Howe in the far east to Discovery Bay in the west. And then it extends into each of our major bays, Port Phillip Bay, Western Port and Corner Inlet, which I don't know if you can see on my mouse, but you'll all be familiar with that. On this map, what you can see are the dark blue areas of the marine national parks and sanctuaries, and these are our strictly no-take marine reserves that were specifically set up for the protection of biodiversity. And then there's these lighter blue areas like at Corner Inlet, just above the prom there, which are our multi-use areas. So again, they were still set up to protect biodiversity, but also to allow for multiple uses like recreational fishing and commercial fishing and things like that. So our monitoring and research has really focused on understanding whether Victoria's marine protected areas have been meeting that purpose they are created for, which is to protect representative biodiversity. And so to understand this, over the past 20 years, there are probably three broad questions that we focused on. And so the first of these has really just been to understand what is actually in our marine protected areas. So when the parks were created, they weren't done with the best available information and habitat mapping, but this was fairly limited. So as managers, we need to understand what's in our parks and where it is to manage it effectively. And so we've undertaken a lot of habitat mapping to understand what's in there. And as well as biological surveys to get a handle on what they are. So the next major aspect that we're seeking to understand is what the condition of the biodiversity is within our marine protected areas. So to do this, we look at the long-term trends of what's inside our park and compare this to what's outside as well. And so that gives us a good handle on the effects of protection and how things are tracking over time. And finally, we also undertake quite a bit of research to understand whether the things that we're managing are being done effectively and if it's making a difference towards protecting biodiversity. So obviously, that's a lot for me to talk about in 10 minutes, and I'm not going to, so don't panic, Cade. So what I want to talk about today is really focus on what's the condition of things in our marine protected areas, what are the long-term trends in abundance, and looking at those inside-out comparisons, and how have the parks performed effectively over the past 20 years. So the main way that we've got this information has been through diver monitoring in our marine protected areas. So we've been really fortunate in that the Victorian government had the foresight to establish diver monitoring surveys before the parks were even created. So these approaches have been slightly modified over the past 20 years, but effectively we've had continuous monitoring of our marine protected areas as well as some reference sites outside of our MPAs as far back as 1998. So this means we have a really good time series to examine the trends in the key species that we're interested in, which is great. So over the years, now hopefully this will play. Give me a thumbs up if it's playing, Cade. Over the years, what we've also done, great, is added new approaches to complement the diver survey. So this footage here is from a technique known as a baited remote underwater video, which some of you may be familiar with. It's basically a camera on legs that we drop on the seafloor with some sardines at the end of this orange pole that you can see sticking out. Fish come along and swim up to it, and we can count the fish, identify what they are. And what we do is we drop these in different habitats inside and outside our parks, and it gives us a sense of what types of fish are using the different habitats, whether that protection is working, what are the differences inside and outside. And we also get really amazing footage from it. Like this is a classic example we got from Cape Howe, this grey nurse shark that we found up at Cape Howe Marine National Park, which we hadn't seen before. Some of the abalone fishes up there have probably seen them more commonly. But yeah, the other thing that we've also been doing is adding some kind of more specialised monitoring to our monitoring programs. So recently with Deakin Uni, we've been undertaking surveys of southern rock lobster, both inside and outside of the MPAs, to understand whether they're being adequately protected. So we've chosen to focus on this species because it's ecologically a really important species. It's a keystone predator on urchins, things that graze on kelp that we want to have there. So really important for reef health. It's also a target of illegal fishing. So it provides that really great indicator that bridges the gap of being ecologically important, needing protection from the parks, and can tell us a lot about the health of our parks and whether things are being effectively protected. So as I said, we're now coming up on 20 years of our marine national parks and sanctuary, and that's meant that we in our team will be thinking a lot about how they've performed over this past 20 years. So next, I'm just going to show you a few key stories from our monitoring programs that tell us something about what our parks have been doing and how species have been tracking in them. There's obviously tons more stories than what I can show you here, so I'll throw in some resources at the end, but these, I guess, are the key ones that I think are the main interesting stories. So one of the simplest ways we can measure biodiversity is through a measure called species richness, which I'm sure many of you here are familiar with, and that's just the number of distinct species that you find in the area that you measure. So what we've recently done is completed a statewide assessment of our whole marine national park and sanctuary network. So basically grouped everything we've measured in together and then compared differences inside and outside of our parks. And what we found was a really nice pattern, which is basically that inside our marine protected areas, we have high richness of fish and also of the large mobile macroinvertebrates we measure. So things like sea stars, abalone, what else do we get? Large mollusks. Those are the types of things we're measuring, feather stars, brittle stars, things like that. And so that high richness is a really good indication that that high level of protection that we give these areas from our marine protected areas is working and having benefit to biodiversity in those areas. Similarly, when we kind of zoomed in and we've looked at southern rock lobster, we found some really great patterns there that show how marine protected areas benefit a species like this. So we've completed these surveys now at Cape Howe, Port Phillip Heads, Wilson's Prom and Point Addis. And at Point Addis, Port Phillip Heads and Wilson's Prom, we found a pretty consistent pattern. The numbers differ between them, but fairly consistent pattern where we get more individuals inside our marine protected areas and they all tend to be larger and often it's significantly more individuals inside. And so this to me is a really nice example of how our marine parks are working and effectively protecting these species, but also some of the benefits of marine protected areas. So something like a southern rock lobster, when it reproduces, it produces little babies, larvae that last for about up to 2 years in the water when they disperse. So that means that some of the larvae from these populations we're protecting in our parks have the potential to effectively disperse from one end of the state to the other. So creating these really important source populations that can then spill over out and replenish reefs outside the parks. So not what the parks were originally intended for when they were designed, but a really significant benefit of having that protection. And so then we've also looked at some of the long-term trends of fish species to compare how they're tracking. And there are some really nice examples. So the blue throat wrasse is a really great example of a species that shows a lot of benefit from marine national parks. I'm sure most of you here, if not everyone, knows this species quite well. One of the most common and widespread reef fish in Victoria. So it's a great one to track. The plots here show the number of blue throat wrasse inside the Marine National Park, which is the black line, and outside, which is the grey line. This is what we call an ecological control chart, and the colours here correspond to abundances that are good. Yellow is like not so good, and red is what we kind of use as a trigger that there's some management that is required. And so at Point Addis and Port Phillip Heads, we got this pretty strong trend of high numbers inside the park, as well as this really nice increasing trend since the parks were created over time. So, a great indication that things are tracking well for that species inside our parks. In the interest of transparency, this isn't the case everywhere. At Wilson's Prom, we don't see those strong differences inside and outside of the park. And in some cases, there has been more outside. But in somewhere like Wilson's Prom, this is an area it's a bit more remote. There's less pressure from recreational fishing, and there's good habitat both inside and outside of the park. So we kind of expect those patterns to be a little bit weaker. But in general, we can see that there's some parks that are having real benefit for this species. And that's also true for a range of other species. So we found this for things like the yellow striped leatherjacket here, the horseshoe leatherjacket, that have those generally higher abundances inside our marine parks. Similarly for things like the scaly fin at Point Addis Marine National Park and the sea sweep. So these are fish that they're all kind of reef attached fish, maybe less so the sea sweep, but they're all pretty reef attached, but they have quite different life histories and feeding modes and things like that. And so across them, we're seeing these kind of patterns of higher numbers inside our parks. And again, not everywhere. At Port Phillip Heads, see the scaly fin doesn't seem fussed whether it's in a park or out of the park, but at Point Addis it does. They do demonstrate, though, that a number of our parks are fulfilling that purpose of protecting a range of fish species quite well. And it's not just restricted to fish. So we've seen similar patterns to what I've described for the green lip abalone at Port Phillip Heads. So again, this is a really ecologically important invertebrate species. And, it's a nice one, again, like the southern rock lobster, that its larvae can disperse outside the park. And so it can replenish those populations outside. But I guess, what was my point here? My point here is not restricted to fish. We're seeing this for large macroinvertebrates like this as well. However, it's not the case for everything that we've been measuring. So in contrast to the species I've just described, we are seeing some declines and signs of trouble for some species. A major example of this is the black lip abalone. So what we have seen in general is a strong long-term decline in this species across the western and central parts of the state where our parks are. We still have often seen high numbers inside our parks. And what we see is that this decline is occurring both inside and outside our parks. So it suggests that this is representing a larger decline than just what's happening inside our parks. And certainly that's been backed up, I guess, by other research that's out there. However, not the case at Cape Howe in the Far East, for example, where things are bouncing up and down. So it's not all doom and gloom everywhere. So it's a complex story, and that's one we're still trying to unpack and understand from our monitoring. And I guess for me, one of the biggest stories that's happening out there and one of the more concerning patterns that we are seeing has been a general decline in large canopy forming brown algal species across the state. From our monitoring that we've recently completed at Point Addis, Port Phillip Heads and Wilson's Prom, where we've looked at these long 20-year time series, we've basically seen a fairly long-term decline that I guess varies in significance depending on which park you're looking at. It's not a it is a complex story, but at Point Addis, it's virtually disappeared from our plots there. At Point Addis, it went through a decline recently and just bouncing back. Sorry, at Port Phillip Heads, and at the Prom again, we've seen that decline. And we followed up on this. This is showing a Colonia radiata, the golden kelp here. But we've also seen this to some extent for Philospora comosa, the crayweed, and these are the two major canopy forming species in Victoria. But Philospora is tracking a little better. But we've also recently used this monitoring and followed up on it with some larger research projects that we've done with Deakin University that are showing that is kind of a statewide trend of decline in certain areas of the state from east to west. And again, not happening everywhere. There are some areas where it's pretty stable and it's pretty okay, but decline is generally the story across the state. In some cases, we know what the cause of this is. It's because urchins have become overabundant and are eating away the cover of this kelp. But in the western parts of the state, we're still seeing this decline. And some recent work we've done is suggesting that this is linked to increases in sea surface temperature that we're seeing in southern Australia. So we're currently working on this and working out how to respond, restore, and kind of conserve kelp in our marine protected areas. Yeah, so those are really the key stories from me. So thanks very much for listening. There's tons more information out there that we're providing. Here are some links, and I think I've sent you these links, Kate, if you're able to pop them in the chat, because I might be able to. So to celebrate our 20th anniversary, we've got a website here. We've developed a webinar series, and on that website, I recently gave a more extended webinar on Victoria's marine biodiversity, and I borrowed quite a few slides of that for this talk. So sorry if you saw that as well, and I've doubled up, but and also more details and the comprehensive kind of findings of these monitoring programs we've been running can be found in the Park Victoria Technical Series, which is a publication that we put out that puts all our data out there and details what we're finding with our monitoring, and that's located here at this website. So thank you very much, everyone. That's it from me.
Awesome. Thanks, Mike. It's really hard when you don't get that applause sort of thing to know that you've actually finished. So that was great. Thank you. There was just one thing I wanted to ask you, Mike, which is in relation to all this research. Are you seeing any large-scale changes in the environment? Like what sort of large-scale changes are you seeing regardless of parks? Like you mentioned a couple there with the algae and what have you, but I guess we're sort of leaning towards like the climate change, and there's these sort of larger impacts that, irregardless of the park itself, it's something that's occurring along our coastline. Is there any sort of indicators or anything that's showing up with that?
Yeah, definitely. Yeah, look, so there's probably three things that jump to mind. One of them I've just spoken about, which is the decline in kelp. The second one, which is related to that, is we're seeing this explosion of urchins, which I'm sure many of you out there diving have noticed in northern Port Phillip Bay. The bay's basically been eaten away by the purple urchin, and on the east coast, we've got Centra stephanus, which is coming in. We're starting to think that the purple urchin increase is linked to warmer sea surface temperatures, which has that climate change aspect to it. And we know that Centra stephanus is coming in from the east where its native range is and extending into Victoria because the East Australia current is pushing in under climate change. And, southeastern Australia is a hotspot for ocean warming. It's one of the hotspots because of that extension of the East Australia current. And what we're starting to see up there is some species that we expect to be there, but they tend to have that more central and western distribution in Victoria. Things like your horseshoe leather jackets, and we're just not seeing as commonly anymore. We just did the survey at Cape Howe, and some of those species we haven't seen since 2015. So we think what we're starting to see is things shifting their range that have that more preference and tolerance for colder water environments. They're starting to shift away. And I guess that's something that's front of mind for us at Parks Victoria, is thinking about, what are these changes that are driven by climate, particularly around species range shifts? How can we manage that? How can we make our parks good habitat for those type of species? And then what's the stuff that we just accept is change that's beyond our ability to manage in the small kind of parcels of water and seafloor that we have? So yeah, it's definitely changes happening rapidly, I think.
Yeah, and that was a tough question to answer. You did a good job with that. We've got a couple of questions here. As I said, we might finish Mike's talk at 8, so that anyone who needs to go, feel free to go, and we will send you around a link to Nicole's. But please, if you need to refresh yourself, go and do that. We'll kick off Nicole's talk at 8. But we've got a couple of talks here. So we've got one from Dave asking, great presentation, Mike. Are there any obvious changes in results of effort that could be attributed to the move from dedicated scientific research efforts to the citizen science surveys or the reef life surveys? Was there sort of a comparison done between the two methods to ensure that the samples are coming back the same?
Yeah, absolutely. So it's a good question. I think I get it properly, but I guess, so a good example of what you're talking about is at Parks Victoria, we have moved, and Cade, maybe you were talking about this, I don't know, when I was trying to get my modem turned on, but we moved from a program that we kind of ran, the subtidal reef monitoring program, and now we switched to reef life servers. So we have lots of rangers at Parks Victoria who are trained in reef life survey. We can also pick up additional data from the citizen science. And what we have done is kind of, what's the word I'm trying to say it in a simple way, but harmonized that data with the reef life survey data. And it's great. I mean, the reef life survey data that we get is fantastic. It's as good as what we were doing in the past, and that's helped us to kind of keep that time series going, because to be honest, cost is an issue for us, and with monitoring programs aren't cheap. And so having that big community that's kind of nationally, it builds more support, it helps sustain the science. It's kind of like the more people get hooked on citizen science and things like reef life survey, the greater the likelihood is that thing will be sustained over a long time. The data's good, we can use it, so we're using it in our monitoring programs. And that goes for things like the Great Victorian Fish Count, like the intelligence and the things we get from that are great. So I'll shut up in a sec, Kate, because I'm going to wrap it on. But just to give you one nice example of some of the work that's come out of this group is, there's been some questions going around about weedy sea dragon distributions around certain areas of Victoria, and there hasn't been a lot of concerted monitoring effort by us and by other, scientists around weedy sea dragon distributions. And so some of the stuff that's come out through the Great Victorian Fish Count, just to show where they are, how widespread they are, where people are picking up, has been really valuable just to know, this is where it is. It doesn't have a restricted distribution or whatever we're trying to find out. So, and what habitats it's found in. So, yeah, I don't know if that answered your question, but I guess in a nutshell, since science is very valuable. And
you got to thanks
great,
Michael.
Yeah.
So I have a feeling in there somewhere that was the answer that he was looking
answer.
for, and the
Yeah.
response. Yeah, and it is a tough one. And so here, look, from one curly question to another. And I think this, unfortunately, is your role as someone who works for state government, is that people like to throw curly questions at you. But the lecturers from universities are fine. Let's ask them the nice, easy questions. But let's get stuck into parks with the curly ones. And you mentioned continuous monitoring, but from my time at PV, it seemed sporadic and irregular. What was the actual frequency? And I guess you can have sites around the state. So is there a regular monitoring, or is it one of those things that, there's obviously been funding gaps and time gaps and different things that are happening, so you're doing the best you can, and the fact that you have 20 years, it helps sort of, I guess it's not so much patch over, but it helps you paint a better picture, because while you may not be monitoring every year, you do have that time series that is often not afforded to anything really.
Well, yeah, it's an interesting comment. Like, I think that, so there have been gaps in it where things haven't been done, but I kind of wonder if that perception hasn't come from the fact that we haven't actually promoted and got out there all the work that we've been doing. Because, really from about 2002 to 2011, there was a lot of monitoring that was done for the subtidal reef monitoring program across heaps of most of our marine national parks, and it was, not annual, but it was close to annual for most of them. Then there was a bit of a sort of slowdown around 2014, and then we picked up reef life survey methods. And so, places like Cape Howe, there has been a few years gap. So, we haven't got it every single year, but we certainly got that, lots of data points across the 20 years, and more than 10, 15 years of data points from that diver monitoring. And then some of the stuff I've talked about, the Bruv and the rock lobster, is stuff that we've only just started, but we've sustained that since about 2016 when all that stuff kicked off. And it could appear a bit sporadic as well, Kay, because I'll just say our model that we use for monitoring, because these campaigns that we're doing now are quite intensive, and not overly cheap. What we do is we focus on one park in one of the marine bioregions. On 1 year, we do a big campaign that year, and then we move to another one. So we're kind of rotating around the state every 3 years. So, you know, it's yeah. I mean, we could always do more monitoring. There's no doubt about that. But you know,
Oh no, and that's, I mean, you have the expertise and the skills in that sort of analysis of data and knowing where your gaps are. So the conclusions you're making are based on knowing what those limitations are. So
absolutely,
yeah, appreciate you taking the time. We've gone past 8 o'clock, but hey, look, we've sort of thrown off the shackles with this weather. I don't know if anyone's hurrying anywhere, and besides, everyone's hung around to listen to Nicole. Because Nicole is the multi-talented, intelligent half of Reef Watch. As my own daughter and others have often commented, all I ever seem to do for work is talk. All Nicole ever seems to do is work. So it's nice to have her to talk. Since being awarded a PhD from the University of Adelaide, she's worked in a variety of fields where she's occasionally had the opportunity to flex her amazing communication skills. She's an amazing artist. This year's logo, last year's logo, the year before's logo, all the Sea Slug Census logos have all been done by Nicole. Our Marine Life of Victoria board game has 64 illustrations, all done by Nicole. And she also makes incredible nudie models. And by that, I mean nudie branks. There's every chance there's probably one sitting on her desk there. She's also a fantastic science communicator and an incredibly awesome person to work next to. I'm honoured to introduce Nicole to bring us up to speed with last year's and this year's fish count.
Thanks, Kate. Thanks for setting the bar pretty high. If I can meet it. Okay, I'm just going to jump right in. I am used to condensing a 15-minute talk down to like 7 minutes or so because I'm usually doing the tail end of the things. I'll try not to speak too quickly, but yeah, as Kate said, if you do need to jump out, I won't be offended. But to kick it off, the dive that counts, the fish count. 20 years ago, some curious divers started making observations of what they were seeing when they were underwater. And this did evolve into a targeted list of species of fish that groups were encouraged to go out and record at a specific time of year. And some of you will recognise this as the Great Victorian Fish Count. There's been a few iterations of the species that we've studied, but pretty soon 25 common and not so common fish, not typically, I guess, targeted by recreational or commercial interests, were selected as the official fish count. And in 2017, we added, I was going to say 10 sharks and rays, but I should probably say 9 sharks and rays and 1 chimera to that list. And what this means is that our citizen scientists, like, they don't need an encyclopedic knowledge of all the fish that exist in Victorian waters. They can just focus on these species. And then we have a consistent list of certain species that we can compare to from year to year. Most clubs have actually adopted, I guess, like a regular site, be it their local marine sanctuary or a pier or something, and they return to it every year. And so all in all, it gives us a snapshot of the fish occurring in places along the Victorian coast each year, and a bit of an estimate of their general abundance. All right, so looking more specifically at last year's fish count, there was definitely a distinct sense of deja vu compared to the previous year, 2020. Many of us were unsure when we might be able to return to the water again about this time last year. The lockdowns for those of us still stuck in Melbourne were lifted with mere weeks to go before the fish count started, but I think everyone did a fantastic job to work within the health regulations and ensure that last year's count went off successfully and safely and responsibly. And from the looks of it, you all managed to actually have some fun too. I'd say that weather was a challenge, and I guess it's quite fitting tonight. We are in for another La Nina year, so it's probably best to expect this bit of the same. So I would say to anyone participating in a fish count, do make sure you're in touch with your local hosts this year so you can be aware of any scheduling changes. What we would say is that it's really great to see that a lot of clubs are registering multiple events. Just to clarify, once you're registered with us, you can actually run as many fish counts as you like within that window. And you may actually appreciate the buffer of having multiple survey dates locked in a year like this. But hopefully I'm all being doom and gloom and we actually have a nice sunny spring, summer. Let's go for that. All up in 2021, we had 46 surveys lodged by 24 groups. It was slightly fewer than previous years, but I think it's still a fantastic effort considering everything that everyone was up against. So let's have a look at some data. In terms of the actual surveys themselves, we would say that there's still a bit of a skew towards surveys being conducted by divers, but snorkelers made up about a third of the surveys undertaken, and there's a bit of a mix, like a few groups offering hybrid events, which is nice because it's still demonstrating that there's appeal for the younger fish counters and non-divers to get involved as well. Interestingly, last year was a bit of a dramatic shift in the location of fish count surveys in terms of the type of marine area being conducted. So in the past few years, about 30% of all surveys were conducted in marine protected areas like marine national parks or sanctuaries. But in 2021, this dropped to about 17%. I was sort of scratching my head about this. We might have to do a bit of a deeper dive, as it were, but we suspect it might have something to do with the fact that we did have an increase in dive clubs doing multiple surveys. And as many of you dive under things like piers and other artificial structures, more than say inside of marine sanctuaries, we think that might have made a bit of a difference. Certainly in the spirit of this year's fish count, we would encourage groups running multiple events to consider comparing their local site to, say, a nearby natural reef or other protected area, because you don't know what you might find. To the fish. For the bony fishes, once again, the blue throat wrasse reigns supreme. These lovely wrasses, as Michael alluded to, they're present across almost all survey locations and habitat types, and they have been for many, many years. This was followed by things like the magpie perch and zebrafish. Zebrafish had a bit of a bump in the overall percentage of surveys they were recorded in last year. Again, haven't really had a chance to get into the data to understand why, but there they were. Both leather jackets, so the horseshoe and the six spine leather jacket that we study, were actually cited kind of proportionally less than previous years. Leather jackets, you can see them under piers as well as on natural reefs. So again, not entirely sure what's going on there, a bit curious. Not exactly explained in that sort of split of location surveys that we saw. For a lot of the kind of, less often cited species, which are all those ones to the far end there, things like red wallongs and boarfish, a couple of the gropers, they tend to show a bit of a starker difference in how many surveys they were seen in compared to the rolling average of the last 5 or 6 years. But I would say that because they appear in far fewer surveys to begin with, it's a little harder to tease apart any trends there. For the target sharks and rays, smooth stingray was observed in the most surveys. Again, pretty much standard for the fish count. It is a common visitor to piers as well as shallow reefs within bays. And obviously it's pretty easy to spot, and some of them can be quite friendly. So no surprises with that one. However, many of the other sharks and ray species we observed didn't appear in the same proportion of surveys as previous years. The draftboard shark was the only one that people saw more of last year. But again, we're talking about a handful of surveys when we get down to these ones. Last year, nobody saw the varied carpet shark, which I'll get to in a minute, and no fish count to date has recorded our little elephant fish or ghost shark. But we might know why now. Obviously, that was really just a snapshot of a snapshot. So if you do want to dive deeper into the rest of the data, you can check out all of the reports that are online. We are a little bit late in getting the report for 2021 up, but please watch this space. Given the data from last year, it did make sense to us to have our varied carpet shark to be an ambassador, I guess, for the hidden gems of our marine environment this year. Well, I guess you could argue that the ghost shark could be the same. The theme hidden gems, I think, works on a few different levels. So whether it's celebrating those little gems that we know as our marine parks and sanctuaries, and I hope that people can recognise at least a few of these and be thinking about the next time they might get to go out and experience those, or the fish themselves. And we are encouraging everyone to keep their eyes peeled for this kind of panel of lesser seen fishy gems during this year's count, as well as any non-target but still equally captivating critters, things like handfish and velvet fish, because you can report those at the end of your survey as well if you do know what they were. And as of course, we're always interested to see whether anyone has seen these guys. We've now heard that maybe we won't see an elephant fish, but we could see a little one. But the other one there is a harlequin fish. This is a southern reef resident that is pretty common elsewhere, but is presumed not to be present in Victorian waters. We do hope one day we might see it. Given that the western blue gropers were actually thought to be absent from Victorian waters until they started showing up in multiple fish counts, who knows? If you do think you have spotted a rare fish, a photo will go a long way to confirm that. So we do partner up with Red Map Australia on this. They've been gathering data on fish outside of their known range for the last 10 years as well. So it's another anniversary. You can upload your photos directly to Red Map. You can include them as additional sightings on the Atlas of Living Australia, or you could put them up for ID on iNaturalist, and that all gets fed into the same system. Gone through all of that. Of course, it is the people who take time out of their schedules each year to explore their local patch that make this project and other citizen science projects possible. So the next few slides are just a celebration of our wonderful fish counters. The literal thousands of you at this point that have taken part since the fish count began. I will say that some photos feature people who are no longer with us. So if anyone thinks they need to duck out for a minute or so, this is just a heads up about that. I didn't want to diminish the contributions of these people and their long-term dedication to this project by leaving them out. On a lighter note, but still a warning, I have delved into the archives to complete this little slideshow. So if you have been involved in the fish count for many years, some of these images may shock you. So for me, the fish count isn't just about getting data on our lesser known species. It's also about opportunities to explore and connect with your local patch of reef, pier or seagrass beds. And most importantly, it's about the passionate citizen scientists, which is you guys, who participate each year and become ambassadors yourselves, I'd say, to kind of, promote the environment and get more and more people involved. So again, I just want to say thank you to everyone who has supported the fish count, whether it's by organising events yourselves as hosts, taking part in a survey or multiple surveys or one every year, as we can see a couple of recurring faces in these photos. Maybe it's standing on shore with barbecue tongs in hand, ready for when everyone gets out of the water. Coming along to nights like this or any other way that you've taken part. You guys really do make this project the success that it is. Got last slide here. So once again, thank you. And now an apology. This is a shameless plug for my other baby, the Sea Slug Census Program and the materials and resources that we've just recently published. Basically, it's just to help Victorians learn a little bit more about their local slug life. If you are interested in getting your hands on a copy, please either get in touch with myself or Cade. There'll be emails up in a second. In terms of the Sea Slug Census Project, if you have spotted a slug or something that you think is a slug between the 30th of September and the 9th of October, so just last week, please upload it to iNaturalist and it will count towards the National Census records. All right, that is it from me. Thanks again to everybody who has supported Reef Watch and the Fish Count for the last 20 years. You're all fantastic. I do hope to see you out there for the 2002 Fish Count. And that's it from me.
Fantastic.